See, okay, so we are on, and welcome to everybody to this new edition of the ILS webinars, the first edition of the new year 2026. We are here, happy to be here with a long-time collaborator and friend, Bernadette Julier from INRAE, so from France. And Bernadette is director of research at let's try my French, at the Unité de Recherche Pluridisciplinaire Prairies and Plantes Fourragères. And today we are going to talk about how to incorporate molecular markers in lucerne breeding, which is Bernadette's area of expertise. So thanks, Bernadette. The mic is yours.
Okay, thanks, Nelson. Thanks to all of you for attending this webinar. I'm sorry my camera is broken. I don't know what's happened today. So I would like to thank the organizer for inviting me to give this webinar. And we will talk today about forage legume, so lucerne or alfalfa, and about molecular markers. So it will be a lot of genetics. I hope it will be interesting for all of you. I have prepared this presentation with my colleagues, Philippe Barre and Marie Pégard. Yes, so molecular markers in breeding programs, in fact, it's a subject on which we already have a proof of concept available on different plant species, and it is already implemented on different major species such as maize or soybean. And in fact, for many years, lucerne lags behind for different reasons. First, because it's an heterozygous species, the plants are heterozygous, and it's an autotetraploid species. So it means there are 4 copies of each chromosomes. And for several years and decades, we have relied on the genome sequence of the model species Medicago truncatula. That was a good opportunity, but we were so happy that we didn't spend much efforts for several years to get the genome sequence of lucerne. And it was also difficult because of this heterozygosity and autotetraploidy. And also because, in fact, the profits of the seed business are not so high on this species. So, and the cost of molecular breeding are expensive. So this was the situation. But we have had recent progresses. I have listed a few steps that were important to go from phenotypic selection to molecular breeding. In 2020, we had the first release of the genome sequence of alfalfa. It was published and followed by others afterwards. Cost-effective and high-throughput genotyping method has been obtained, that is GBS. It's not dedicated only on alfalfa or lucerne, but it works on lucerne, even with this genetic complexity. We have shown that it's possible to make a genotyping at the population level, so to obtain allele frequency. And it's very important because it means that it's possible to reduce the genotyping cost when you want to work at the population, so at the variety level. We have refined the method, the GBS method, to lower missing data because we had different series of data with a lot of missing data, and it was really a problem. And there have been several tests showing that genomic predictions were very promising. Just to remind you, if you are not familiar with lucerne, the varieties are synthetic varieties, meaning that at the beginning of the variety, you have a polycross with a certain number of parents, minimum 4 and maximum a lot. And these parents are polycrossed, so intercrossed, during several generations, 3 or maybe quite often 4, to obtain the variety, so the commercial seeds. So it means that each variety is a large family with many plants, many genotypes. All are different, but they are related. Okay, the traditional breeding methods are, yes, a type of breeding scheme. There are many different versions, but typically it's this type of scheme. You introduce new origins in your breeding pool. You study the plants in the nursery of space plants, and then you choose individual plants on their phenotype. It's for 3 years. You harvest seeds on the mother plants you have selected, and you test this progeny in microplots for 3 more years. From this result, you go back to the individual plants if you still have them, or you go back to the seeds you had harvested before, and you continue with the polycross of these best plants. And the progeny of this polycross can be evaluated in a multi-site trials, and if it works, you obtain some new varieties, candidate varieties, and this population is also an input into your breeding pool. So it's quite long because, in fact, because of the time of testing, and testing is at least 3 years for this perennial species. So why using molecular markers in this breeding program? So there are different possibilities. First, to describe genetic diversity. Lucerne is spread over all the world, and so there is a lot of genetic diversity. There is a lot of genetic diversity within the variety. So it's a way to manage the genetic diversity. It can also help to improve the distinction between varieties. I will not discuss about that. All the topics in gray, it's just to remind them, but I will not discuss about. And also to decipher alfalfa expansion history in the world. The markers can also be used to identify genes or locus involved in quantitative traits through association genetics or before mapping populations. So you make a correlation between genetic and phenotypic data. And they can be used for allele mining in candidate genes. So if you have candidate genes for any traits, you can look at the diversity in these genes. And the markers can be used to predict the phenotype of one individual from its genotype with a calibration setup on a training population. So that is genomic selection or genomic prediction. So there is a general scheme here, and I think you are more or less familiar with that. So you have a breeding population, and you genotype, phenotype the plants. You obtain a prediction model that is applied on your candidate for selection. And it's useful to obtain once you have the genotypes of your candidates, you can predict their phenotypic traits without any evaluation in fields or tests. Sorry. Okay, so for many years, in fact, we have had ideas to work on Lucerne with markers, but we had not much fun at the European level, at least. And recently, for the last almost 10 years now, we have been able to obtain funds from the European Union. So the first project I was coordinating was called UCLEG. It was the title was Breeding Forage and Grain Legumes to Increase EU's and China's Protein Self-Sufficiency. And it lasted for 4 years. And we had 26 partners in 13 European countries and 12 partners in China. We worked on 5 species, among which lucerne. And the general objective was to develop efficient breeding strategy for legume crops and improve crop diversification, crop productivity, yield stability, and protein content or other quality traits. And we have based our analysis on the exploitation of genetic diversity, either genetic resources or elite material, meaning varieties. Yes, here. And we have carried out phenotypic evaluation, developed molecular markers when needed for this species, and it was the case for lucerne. We have genotyped this population, and then we have studied the diversity, the genetic determinants of the main important traits, look for associated markers, and tested genomic selection. And we have spent efforts to develop statistical methods and to spread them among the partners. So this was conducted also with breeders and with the final aim to improve varieties. And the second project is ongoing, again on genetics of legumes. It's called BELIS, for breeding European legumes for increased sustainability. It attacks, in fact, 4 lock-ins that explain why genetic progress on legumes are not so high. First, there are genetic lock-ins, so we need more knowledge. There are some regulatory lock-ins, some recommendations. So even when, so regulatory is mostly about registration. Recommendation is even if you have registered varieties, the qualities of the varieties are not spread towards the farmers. And there are also some financial lock-ins, meaning that the possibilities, the support the companies or breeders can afford on legumes are relatively low. So we are working this project on the development of tools for legume breeding to help the whole process of variety registration, production and registration, and their use by farmers. So also working on the registration process to improve the process, be more efficient, improve the evaluation and recommendation, and also improve the business and government models. We are working on 14 species, among which lucerne, and we are also developing a platform to disseminate the results. And we have built the BELIS network that is open to anyone who would like to know more about our results and contribute to this project, to listen about this project and to maybe to work together in other projects in the future. Well, the scientific aspect of my talk will discuss about the panel of populations we can use for genetic studies on lucerne. Very often, in fact, the strategy is to study panels of individuals. And this is probably related to the fact that when you work on other species that are, for which you have pure lines, it's very easy to work at the individual level, and the individual and the variety level is finally the same. So very briefly, in fact, the panel of individuals are obtained from a population that can be the progeny of a polycross. And on these individuals, you do genotyping. You can have some phenotyping studies, but they are very limited because each plant is alone. So you have a single plant and there is no possibility to multiply it except via cloning, but it's very laborious. So if you want to study the phenotypic value of these individuals, you have to obtain progenies and make the phenotyping on these progenies. So this is feasible and it's done, but it's quite labor intensive and not very well related to the breeding process, I would say. And another strategy is to work on a panel of populations. You have a series of populations, so it may be varieties, registered varieties, breeding material, land races, any type of population that is more or less a variety. On these varieties, on this population, you can have genotyping. In that case, you genotype the pools of DNA. So meaning that you obtain the frequency of alleles. And you can do phenotyping And as you have seeds, and you can multiply the seeds again, you can study traits at a real level, I would say. So you can have field trials with dense canopies. You can have test for resistance, disease resistance in control conditions. You can multiply the number of traits you can study. And with this data, you can study diversity, relationships, so kinship between the population, association genetics, and genomic selection. And our hypothesis is when you identify some genetic determinants, by using this panel of population, you can use this result for selecting plants at the population. So you switch from the populations you are studying to the selection of plants. So it's an hypothesis that this was really an hypothesis. So we started with collecting a panel of 400 populations and varieties. We obtained genotyping data on 395 populations, mostly cultivars and 22 land races, and they originate from all the worlds, but mostly from Europe. And the constraint was that the dormancy range was between 3 to 7. So with this material, we developed genotyping. We genotyped them with GBS markers, and we obtained quite a large number of SNP, more than 200,000, with a very low number of missing values, and this was our objective. So it means that the level of imputation is very, very low. So we work on the true values. And the marker density over the genome was very, very high. With these markers, we have done a PCA, or principal component analysis, with the populations. And here it's a result, each point is a population on the PCA, and the ellipses are based on the DAPC clustering. And we have obtained 7 groups, a very small group of 2 populations that have some Medicago falcata genetic background. One group, number 6, with Chinese origins, that is completely different from the other population, and a large group of either European and American accessions that was split into 5 groups, overlapping groups, but from one to the other. The first group in blue was from France and Northern Europe. The second was Southern and Eastern Europe. Then we had the group 4 with European and American accessions. Group 5 with only USA accession plus 1 Chinese accession. And group 2 with a population from North America. So in fact, there is, yes, an overlapping of these groups, but you see a difference between European accession and North American accessions. Even if lucerne originates from the ancient world, so we could have expected that the American accession are more or less included within the European ones. So we don't know the reason for this difference. Maybe evolution, natural selection, or human selection or drift. Anyway, the FST between groups were very small, but this is very common to lucerne. Here, it was another aspect of the study in which we have collected accession from breeders, different breeders. They were just asked to provide some accession from their breeding pools. So I don't know exactly if they provided something that was the hearth of their breeding pool or a large range of diversity, but it was very interesting to see the difference. So we had breeder 1 that had a large range of diversity from European and maybe a little bit of American diversity, but not the extreme of American origins, and no trace of origins from Falcata or Chinese, except one accession. Another, the breeder 2, B, had a population really in the middle of the North French and Northern origins, so a very narrow genetic basis, in fact. And breeder D had accession of breeding material from Europe, North and East, South, etc. And some populations that were probably close to the Falcata types. So this breeder has probably enlarged his breeding pool. Okay. So I would like to present you some results on the markers associated to trait variation. On this population, we have had trials established in 2 locations, so Lusignan in France and Novi Sad in Serbia. And we have phenotypes traits related to the agronomic value and quality traits, so phenology, autumn dormancy, dry matter yield, acid detergent fiber as a component of digestibility, and protein content. And we have scored them during 3 years with 4 to 5 cuts per year. We have obtained phenotype, and at least in France, our trials were quite heterogeneous with special effects. So we adjusted this variation for we adjusted the data, the trait for the microenvironmental variation inside each trial. And we also adjusted for year and location effects. And we proceed with GWAS. And here, for example, with the protein content, and we obtain a few QTLs that contribute to explain these traits. And an example on the right-hand part with a marker that explains 10% of the variation. So when the frequency of this marker increases, the protein content increases a little bit. So this could be markers to be used for breeding. We have also scored the resistance to one important disease, anthracnose, in tests carried out under controlled conditions. And we have obtained an interesting situation where we had almost 60% of the variation explained by QTLs, with one major QTL or two, in fact, 2 important QTLs on chromosome 8. It was interesting to see that we had 4 QTLs on chromosome 8, at the bottom of chromosome 8. And they are close, but they are different, definitely. And we had also a marker on chromosome 4. And just for information, this marker is very close to a QTL found on Medicago truncatula for the resistance to this disease. So on Medicago truncatula, it explained a lot of the diversity, the variation for this trait. And in lucerne, it explained just a little bit. And from some of these QTL, we had genes, the markers, the SNP were included in candidate genes, and some are leucine-rich repeat domains from this LRR family. Okay, and what about genomic selection? Genomic selection on this population here is presented for dormancy, autumn growth, and yield, on autumn yield. Just a view with GBLUP. And we obtain quite a high predicting ability, depending on the traits, between 0.84 and 0.8. Yes, here we tested different methods, but they are more or less, they give similar results. And we had this promising result for these traits. And yes, this has been published. And we have done the same. Yes, sorry. And here we have studied the effect of the training population size to see if our population was sufficient or not. And here you can see that you obtain, in fact, quite a high, the maximum predicting ability after when you have about 100 populations, but you decrease the standard deviation when you have more. So when we add the maximum number of population to 217 here, we had the highest value of predicting ability and very, very low standard deviation of this evaluation. So it's very, very promising. We have done the same for forage yield and quality. So here, dry matter yield, ADF content and protein content, and again, very, very interesting levels of predicting ability over 0.5. And so this was done on adjusted traits over location, years, and cuts. So I hope it's very, very good phenotyping values. And the same here for anthracnose resistance, and we obtained the highest predictive ability over all the traits with 0.85. And again, we obtained this result after more or less just a little bit of 100 accession. But when we have more than 200 accessions, then the predicting ability is very high, at the maximum, and the standard deviation is very low. Okay, and so we have used these results to calculate genetic gain we could have with genomic selection. So the estimation of genetic progress with phenotypic selection is the equation of the breeder. And the same equation can be produced with genomic selection, in which you introduce a correlation between the estimated genomic yield and the measured yield. And of course, the time is not the same as with phenotypic selection, because you can expect quite a very quick genomic selection. So here we have used the result of our trials. So we have heritabilities that were not so high in our situations, the phenotypic standard deviation from our results, the predictive ability obtained for these three traits, and the calculation of the genetic gain per year with phenomic selection and with a genomic selection. And there is a factor 6, more or less. So meaning that the genetic selection could be 6 times more efficient. So this is very, very interesting. And a large part of this result is due to the reduction of the time needed to achieve this selection. So what now for breeding? I think the first thing is to go to the proof of concept. So we have established a protocol with French breeders who were volunteered to test genomic selection in comparison to phenotypic selection. So they decided to choose a nursery of about 800 plants, space plants in the nursery. So they go from one side with phenotyping selection as usual. They selected the best individual from their point of view. And the idea is to make polycross. And on the other side, we genotype these plants, predict the genomic value of these plants, and if possible, predict the genotype of these plants for the QTLs. And again, they receive the results. They select for the best individuals and that are polycrossed. So these are based on the list of QTLs and the genomic prediction we had obtained before. So how it works? So for phenotypic selection, they choose plants from their plant height, lodging, vigor, disease resistance. And for the marker-assisted selection, in fact, when we have genotyped the plants, we have obtained much less markers. But yes, so 86,000 markers. But in fact, the predicting ability accepts a low number of markers. It's here an example on the right-hand side on the figure, that even if you have just a part of the markers, a few thousand, 10,000s of markers, the predicting ability is still very high. So in fact, it's not really a problem to have less markers, or this number of markers, not 200,000, but 86,000 markers. So we predict forage yield, forage quality, so ADF and protein content, lodging, anthracnose resistance, and autumn dormancy. So the breeders add this data set. For the QTL, unfortunately, no markers were available for the QTL we had because of this decrease of number of markers. And each breeder makes his or selection. So here are the 4 breeders, and so the process for each breeder. So 2 breeders decided to split their efforts in 2 type of material based on the dormancy range. And here how it works. So now this year they have obtained the seeds for all the polycross, here for phenotypic selection, and here for the genomic selection. For the genomic selection, in fact, they collected leaflets on the plants and they sent these leaflets to us, where we proceeded to DNA extraction and GBS sequencing. And this was quite interesting also from a practical point of view to see that the process is not so easy. If you say, yeah, you can hope that with the markers it's very easy. Well, it's not so easy because from time to time you have some problems with DNA extraction, with libraries, with sequencing. So, but at the end, so we were able to give the predictions for these plants, and they have chosen the best plant from their point of view. And again, the seeds have been harvested during this summer. So what are the next steps? So next spring, we will have the sowings. So at the end, we have 12 experimental varieties. We will add 4 controls. So 2 from adapted to the northern part of France, 2 to the southern part of France. Let's say in a neutral location, that is INRAE. And for each breeder, they will test their own varieties plus the 4 control varieties. And so the traits to be scored will be, again, forage yield, forage quality, lodging, anthracnose resistance. And the idea is to compare the experimental varieties to the controls, of course, and to compare the phenotypic selection to the marker-assisted selections. And we hope a lot of progress, and it's, I think, important to have this proof of concept on a real scale. Small scale, but real scale. The next step, in my opinion, is to be able to make the genomic prediction equation evolve. Probably not to improve the predicting ability, because we have seen that we have enough populations in the training population. So it's not because you add more populations that you obtain better results for the predicting ability, but it's mostly to increase the genetic diversity, to extend this genetic diversity, but also on the other side to adjust this diversity to the breeding pools. As I've shown before that depending on the breeding pools, some have a very narrow breeding pool. So maybe if you have an equation established on a large breeding pool, your ability to predict the diversity, the genetic value on your specific breeding pool, it's not so good. So so it's why we would like to test this evolution of the genomic prediction equations. So to do that, we have established new trials with new material. So we have established a trial with 50 varieties, including 4 controls in 2 locations. So the 2 same locations as before, Lusignan and Novi Sad. So these are registered varieties. So in fact, recent ones are varieties that were not included in the last last sample of populations. And at each breeder's place, so the 4 breeders again, they have established a trial with their own material, their experimental material, on the 4 same control varieties. So the trials have been sown in spring 2024, and again, the same traits are scored. So we have now 1 year of result. We are doing genotyping with the same GBS protocol, and now the work will be to merge the data set and to have statistics on this extended data set. So as a conclusion, I hope that I've shown we are able to revise the alfalfa breeding scheme. So we have a renewed view of genetic diversity. We were expecting a genetic diversity clustered by dormancy range, and we have a genetic diversity clustered by a geographic range. We have been able to identify major QTLs, so it's very interesting for progressing on specific traits, especially when there are oligogenic traits. We have obtained high predictive ability with a genomic prediction, and we have calculated a high potential increase of genetic gain per year. So we could imagine to revise this genetic breeding pool. To revise this breeding scheme. Again, we have the breeding pool and the introduction of new origins, and we could start with growing plantlets and extracting DNA at the very, very beginning of plant life, make genomic predictions and score the plants for the allele diversity at the QTL. Maybe it could last for 6 months if everything works correctly. Then you can predict the genetic value of the plant and select the best plants and go directly to the polycross of the best plants. So it could be very, very quick. Then you go with evaluation in multi-site trials, and here you have evaluation of new populations that could be introduced again in the genomic prediction equations. Okay, so my idea was to, starting from the panel of populations, we studied first, then I hope we are able to add new population along years from different breeders. And we continue with genotyping and phenotyping. Of course, we have to make sure that we use the same methodologies. And with appropriate statistics, of course, we are able to update the view of diversity again, make new calculation for with association genetics and with genomic selection. And of course, we need adequate experimental design, for example, to be sure to have control varieties and to have the same genotyping methods and be able to merge different series of phenotypic and genetic datasets. So, of course, these concepts still have to be proved with a measurement of achieved genetic gain within breeding pools, meaning restricted diversity with the integration of QTLs. And so this is ongoing with the proof of concept work. And of course, the cost efficiency still has to be optimized in my mind. For that, we need to produce genomic predictive equation and QTLs for all the breeding traits, because it becomes very efficient if you are able to predict many, many traits in a single genotyping effort, I would say. And we also have to lower the genotyping costs for sure. Okay, so thank you for your attention. I have thanks a number of collaborators and funding, European funding mostly. Thanks.
Thank you. Thank you, Bernadette. It was a pleasure. It was a very interesting presentation. Now it's time for our audience. If you have any questions on the topics, I also take a look at the chat. There's somebody
Yes, I
writing.
will be glad to answer questions, if any.
Sure. If nobody raises their hand, I will start just to break the ice. And so it's I don't know if it's too technical, but you showed in two slides that on one side that you work with very low, well, medium low heritabilities on your traits, 0.2, 0.3, which is part of the game with alfalfa, and then that you also are able to achieve medium high predictive abilities measured as a correlation between true and predicted phenotypes, 0.6, 7, 8, depending on the traits. And so that's a bit surprising. How do you reconcile that? It appears that the model is able to go well beyond the limits of heritability.
Well, I'm not sure to be able
if it's too
to.
technical, it's okay, but it struck at me, so I just wanted to know if.
Well, I'm not sure to be able to answer. For sure, we are calculating heritability and predicting ability at the same level. So it's not, let's say, a technical problem in which we would have heritability at a cut result, for example, and a prediction at adjusted results. I have to think about this, and I'm sure it's a very good question, and I should have asked myself the same question.
It was striking. I'm not putting in doubt, potentiate your work, which I know very well, and you're working very well with it. But that's surprising and
yeah, sure.
a pleasant surprise. Okay, let's see if somebody else from the chat is raising their hands. Doesn't appear so. I'm thanking again you for your presentation. It was super interesting and also a refreshing recap about all the things that are happening on alfalfa, which is a very difficult crop to work with. And I thank you again, Bernadette, for your contribution. To everybody, thanks for being here. That was the first webinar for this year, and we have still 3 to go, 4 to go, something like that. And as usual, and also I thank my institute, CREA, which is allowing us to use the actual infrastructure to record and share everything. And as a last reminder, our conference is coming up. The ELS5 conference is coming up. There's still about a couple of months for submitting your abstract if you want to participate. Or and then there is, if you just want to participate to the conference, there is the usual pricing scheme with early bird and the things that you are used to. Okay, thanks to everybody, and see you in a month.
Thank you a lot. Bye-bye.
Bye. And I'm stopping the recording.